Welcome everyone, I'm Ben Peacock. I'm presenting today on NanoFCM's NanoAnalyzer platform, a way to comprehensively understand LNPs from a single particle measurement. This is looking at the physical characterization, as well as the cargo information and even surface properties from any targeting modalities we might be utilizing. I'm going to start this webinar by just describing some of the qualities of our technology that really allow for such high sensitivity measurement from the single particle perspective. We're then going to quickly just jump into some case studies, and I want to use these to demonstrate how all these different measurements and all these different parameters that we could come to understand about formulations can impact the way we generate nanotherapeutics. So this is going to first look at empty loading ratios in LNP formulations. I'm then going to look at how we actually measure mRNAs in single particles and quantify the number of copies per particle. And then we're going to talk about these surface functionalizations, whether it's targeting ligands, antibodies bound to the surface, how we can really quantify this from a single particle perspective. And then I just want to demonstrate how nano-flowcytometry measurements line up with other measurements in terms of size and encapsulation efficiency, and describe where it can be utilized in the workflow for nanoparticle development. So I'm going to show a quick video here where we're going to look at what it's like for a particle to go through the system. And the first thing I want to mention is that we're utilizing only sort of 10 microliters of a sample. This is a very low volume that we're loading into the system. And what's happening from the particle perspective, these are being aligned one by one to pass through the center of the laser. With each interaction, we're collecting data in 3 channels for each single LNP. The first thing we collect is size scatter data, and this actually relates to the size of the particle, meaning we can count the particle as it goes past and get that sizing information. The red and green fluorescent channels mean that we can understand things like the RNA cargo presence, and we can understand something about the surface, like these targeting ligands. So we can see here just a still image of the what it's like for these particles to be going through the system, or what it's like for us to see them in the software. We can actually see particles down to 40 nanometers in diameter, so we've got really high sensitive detection in the size scatter channel. And when a particle, whether it's labeled or not, goes through the laser, we will see it in that top event burst trace there, with the intensity relating to the size of the particle. Now we also have incredible fluorescence detection, so we can see things like a single PE molecule. We can see a single labeled mRNA. And what this means is that when the particle passes the laser, we can have simultaneous events in 2 or up to 3 channels for the size in blue, and for something like the RNA in the green here. And this means that we're able to separate out these populations in a very binary way. These particles are either considered loaded and have a fluorescent signature, or they are unloaded and have no fluorescent signature. So a lot of the things that I'm going to describe, it's going to be a lot of data to run through, but all of this can be attained in the same 1-minute measurement. And we start off always by looking at these physical characteristics of the size and the concentration. We can see here, we get the size distribution built up of thousands of measurements in that single-minute runtime for particles throughout a range between 40 and 200 nanometers in this instance, but really goes up to 1,000 nanometers. I've included just an image here at the bottom of the screen as well, showing what it's like for 4 distinct populations to go through the system. So when there's this cocktail set of 4 different nanosphere populations just of silica, we can see really nice distinction between these different sizes. And these can only be between 20, 30 nanometers in difference, but they're really nicely resolved in the system. When it comes to actually utilizing the fluorescence, the ways that we can label either the cargo or the surface are really flexible. One of the main things that are utilized are membrane permeable nucleic acid dyes. So we don't have to work with pre-labeled material. We can just do a quick incubation, sort of a 30-minute incubation that allows us to see all the RNA in the system, whether it is inside a particle or if it is free. So this means we can work with siRNA, mRNA, saRNA, all the different types that you might be working with. It's a simple, fast incubation, and means that very small quantities of your sample will actually be required. So I'm going to show just two side-by-side examples. And the first one you can think of as a control, just to demonstrate this process is really working. If we take a formulation which has not included any mRNA during formulation, we see that there's no fluorescence in the system, even though it has been incubated with the dye. The dye really requires that mRNA to become fluorescent. And so what we can see here is that once we have that proper mRNA LNP formulation, we see really distinct identification of the loaded population. And of course, each of these populations will have their concentration analyzed, and we'll get size distribution graphs for each of these. So if there are significant differences between the sizing of the populations, we'll also identify them. And again, I really want to emphasize this, but this can be, I'm going to talk about the quantitation of the RNA copies, but this would be attained in the same time as measuring the loading and empty ratio. So here I've bisected the dot plot in left and right instead of top and bottom. On the right, we've got one of the populations that we keep describing. It's the LNPs that are containing our RNA that has subsequently been made fluorescent by a nucleic acid dye. On the left, though, what we've got are single mRNA copies that are fluorescent and are going past the laser, providing the fluorescence that we can use to quantify our loaded RNA population. In essence, just taking the average of the single mRNA population to understand what the fluorescence of a particle really means in terms of copy number. From this, we can start to describe all the particles in the system in frequency distribution graphs or event distribution graphs. So we can see here the average copy number in this formulation will be 1 or 2 copies, but we can really see the full range as well that is described using the NanoAnalyzer platform. The last thing that I want to talk about is, again, something that can be achieved simultaneously, and it's the surface ligand measurements. So we can expect that there'll be a range of particles displaying different quantities of any ligand that we want to display. Maybe it's an antibody that you want fixed to the surface, or maybe it's a ligand that's built in during formulation. And to describe these, we actually have MESF earth beads that can convert this fluorescence unit into number of antibodies or number of ligands presented on the surface of these particles. And the relationship between fluorescence and the number of units is very linear, as you can see in this graph here, and that we have very nice, robust protocol for this type of quantification. Now, there's multiple ways that we can actually look into applying this fluorescence. So we'll quite often work with antibodies or nanobodies or aptamers, which will incubate with our formulation, or we can work with ligands which have directly been conjugated to fluorophores, so that during formulation, we have got everything integrated and ready to go. And we've discussed a little bit previously how all these parameters can come together. So the first thing we'll get would be that absolute quantification of the number of ligands on the surface that are presented by these particles. But we'll simultaneously get that size measurement so that we can describe the surface area and thus the ligand density of these particles, describing those which have maybe just a few ligands on the surface, all the way to those which are very well decorated with our targeting moiety. And so we can describe this in these frequency histograms that I keep coming back to, because it's a fantastic way to describe everything that's happening in your formulation. So with that, I just want to start getting into some real case studies and showing you how the data really comes out. So in the past, we worked with VAX equity, so working with a group looking at self-amplifying RNA. And the research question which was presented was, do formulation parameters and anything about these particles, because we understand saRNA as a very large molecule that can allow it for increased protein expression, but that the size of this RNA molecule could mean that encapsulation and formulation dynamics can be slightly different. So a panel was designed focusing on the NP ratio. So this NP ratio series goes from 2 to 14. So this is changing the balance between the number of ionizable lipids and the number of RNA, essentially, like a simplified way of viewing this. And we've done this with 2 different formulation designs using 2 different ionizable lipids that we'll call lipid 1 and lipid 3. Now, the first thing that we can take away from this research is that the conventional means of measuring these formulations didn't really identify any changes across the NP ratio series. So we can see we're increasing from left to right the NP ratio, but the size and the PDI done by the DLS and the encapsulation efficiency done by the Ribogreen assay really didn't identify too much that was happening. We see very different things, though, when we actually do advanced characterization. When we look at the empty loading ratio, we can see a really nice relationship that as we increase the NP ratio, the proportion of loaded particles starts to decrease. Now, there's two really cool ways to look at this. We can discuss these formulations now from the proportion of particles that are loaded that actually are going to be actively delivering our RNA therapeutic. Or we can describe the empty particle population as the contaminant population, or even just a waste of resources. When it gets to the full upscaling processes, this could be an incredible waste that is being put into these formulations. And of course, what's really informative is how this might relate to in vitro and in the future in vivo studies. So we can see here that the luciferase response assay for the formulation made with ionizable lipid 1 shows as NP ratio increases, we actually have this reduced luciferase response in line with the empty loading ratio changes. I will highlight that obviously this didn't occur for all the different panels. So ionizable lipid 3, these formulations didn't show the same response, but it's still incredibly informative data as we move forward. So let's talk about some quantitation. Let's look at an example utilizing mRNA. So this was done in collaboration with the PNI, utilizing just 4 different NP ratios in this set. But again, when we first look at the conventional measurements for these EGFP mRNA LNPs, we see very little change in the sizing by DLS. We see very little change in the encapsulation efficiency by Ribogreen. And interestingly, for their specific formulations, we didn't see too much impact of the NP ratio. Again, I think this is quite specific to what they were doing. But what we did see was a real change in the quantity of mRNAs that were encapsulated. We see here 2 of the dot plots for NP4 and NP12. And I used the dashed blue line here to demonstrate that on the NP4 formulation, where we've got like that increased ratio of the ionizable lipid to the RNA, we've got multiple particles with greater fluorescence and hence greater mRNA contained. If we look at this relationship across all 4 of these formulations, we see a really nice relationship between the NP ratio and the median copy number. And again, I really love seeing these frequency histograms that help us to understand what's really happening. We again here describe that the NP4 particles, yes, that there's a number of them that contain considerably more RNA, but we've actually got some data here that shows that the NP12 population has something really good going for it as well. It's quite a homogeneous distribution, or at least there's little heterogeneity in the RNA quantity per particle, which could also be described as a benefit. There's very little difference in the amount of RNA that will be delivered between each of these LNPs. And again, just to highlight that multi-parameter nature of the NanoAnalyzer platform. And something I just want to do is highlight the multi-parameter nature of the NanoAnalyzer platform. So we're going to look at the relationship between the size of particles measured and the quantity of RNA they contain. We've just described the NP12 population as having a nice sort of homogeneous distribution of RNA. But what we can see here is that there's very little relationship between the size of the particles and the number of mRNAs in each particle, rather. It's a very different story for the NP4 formulation, where we've got that abundance of ionizable lipid, so that during formulation of these particles, we actually have really nice incorporation of the mRNA, and the larger particles are able to encapsulate more RNA, highlighting that there's a considerable degree of control in these formulations and giving us a really much better understanding of what we're generating with these different parameters. We can see this presented really nicely here as we try and understand what's happening across the full range of NP ratios here, where we got a really nice relationship. As we increase NP ratio, the correlation between size and copy number starts to decrease. So the last case study I really want to get into is looking at these surface ligand analyses. And we were lucky enough to work with 4Base Bio. So that they have a platform, the Hermes platform, which helps to integrate and incorporate like targeting or non-targeting ligands. In this case, we were able to utilize pre-conjugated fluorescent ligands. So they have the electrophorate already on the ligand during formulation, meaning that when we get to quantification, it's really nice and simple. But we can, of course, do this with any of the previously mentioned labeling protocols. So all the data in this case study is all done on the NanoAnalyzer. So we first looked at the concentration. That there are, of course, differences, but maybe not too informative just at this point. When we look at the particle sizing and something similar to PDI, utilizing the average particle size and the standard deviation, we see very little difference between these 4 formulations. And when we start to look at what I'll term the presentation ratio, so the proportion of particles that present our targeting ligand, we can see 3 formulations really stand out. These have over 80% of their particle presenting our ligand, whereas formulation A, we can see that less than half are actually presenting our targeting ligand. I described briefly before how we can start to quantify this using the nano-FCM earth beads. And we see formulation A and B. Those particles which do present the targeting ligand actually have fewer per particle than both C and D, which has a nice enriched particles that on average have about 100 of these targeting ligands presented on the surface. And of course, you can probably guess what I'm going to show here, these frequency distribution histograms that combine those surface area measurements based on the diameter measurement and the counting of those surface ligands. So we get to look at these 2 very different profiles that have basically identified that we have formulations C and D as our front runners to take through to any in vitro and in vivo studies. So lastly, I'm just going to look at some data demonstrating how we relate to more normal measurements. We've described the advanced nature of nano-flowcytometry, but the simple physical measurements that we get, such as the sizing, align very nicely with things like cryo-EM. So we can see here a liposomal formulation that has been measured by both cryo-EM and nano-flowcytometry. And we can see that the size distribution histograms presented by both techniques are very similar. The main difference being that the nano-flowcytometry approach provides measurements of thousands of particles very rapidly, as I keep saying, that 1-minute runtime. Another thing that I'll just quickly compare to is the Ribogreen method. Many of us who have to use this aren't particularly keen on using it. But what we can do instead is utilize nano-flowcytometry, measure those populations that we see on the left. So in red, the LNPs with the RNA, so the encapsulated RNA population. We can measure the free mRNA population that's not associated with the particle. And once everything has been enumerated, we can describe the encapsulation efficiency, but from a single particle perspective. And even though we've taken this measurement from single particle and single mRNA nature, it aligns very nicely with the bulk measurement from the Ribogreen kit. So this is a really nice way to describe how effective the NanoAnalyzer platform can be. It's not only orthogonal to some of the key techniques that we all utilize, but it's also providing advanced measurements simultaneously. And so this means the NanoAnalyzer has been utilized at multiple stages in nanotherapeutic development. At the very beginning of in research and design, during screening, we can see obviously many benefits to utilizing the NanoAnalyzer system. I think we see most benefit maybe during sort of the down selection processes, where we really want to identify what to take forward using the advanced quantitative data we can attain by nano-flowcytometry. Some of these advanced characteristics that we measure as well can really help identify any changes that might occur during upscaling processes that may not be picked up by conventional techniques. I really want to highlight as well that if there is something that you want to show about your product, such as the efficacy of your targeting ligand, such as the efficacy of your ability to load lots of RNA, then using something with advanced characterization qualities like the NanoAnalyzer is going to really let you stand out. And of course, we're starting to see, because we can utilize CFR Part 11 compliant software, that we can start to introduce nano-flowcytometry into even more parts of the workflow, such as in QC and GMP environments. And we can really start to monitor what's happening to these LNPs in storage and transportation as well. So I hope I've really brought attention to the speed of these measurements. And the fact that you get so many different qualities analyzed simultaneously, including the concentration, the size distribution, and all the different cargo and surface qualities that are going to be really important to describe your formulation. This is, of course, a platform that is ready to be utilized in both academia and industry. We see lots of interest from both research to manufacture and production. And you can reach out to us through our email address, through our website, and you'll find that I do a few of these webinars across different nanoparticles, such as EVs and viruses as well. Again, it's an incredibly flexible platform. With that, I'd love to head into Q&A and just answer any questions focusing on LNPs. Thank you so much for listening, and I'll answer your questions soon.
I'd like to thank our speaker for an informative and interesting presentation. We're going to begin the live Q&A session in just a few moments, so please go ahead and submit your questions if you haven't done so already. Before we start the Q&A, a reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So please do keep an eye on your email and share the link with your colleagues who might not have been able to tune in today. All right, we'll start the Q&A now. And welcome everyone to the Q&A. Thank you for being here. Ben is ready to take your questions. Hope you enjoyed his presentation. Just wanted to remind you that if you have a question at any time, you can enter it in the Ask a Question panel on your screen. We're going to be taking questions live for the next several minutes, so please get those questions in. Ben, thank you once again for that presentation. I'm going to dive right into the questions, and I'm sure others will come in as we get started. The first question I have for you from our audience is, what is the specific workflow to be able to quantify the number of mRNA copies per particle using the nano-FCM, including the controls that are used?
Yeah, this is a great start. So there's a couple of different ways that we can work, but the basic principle that we're going to take is that once the nucleic acids are labeled, we can convert that fluorescent intensity of a single copy as it goes past the laser to like a relative unit for like a single copy in an LNP. And so by taking either a controlled sample that is just unencapsulated mRNA that has been labeled, or by isolating that population with the software in a mixed LNP mRNA sample, we can take the average of those fluorescent units and prepare that as like the base unit for assessing the quantification of the LNPs themselves.
Excellent, excellent. Thank you so much. Next question I have from our audience. How much assay development needs to be done for different lipids?
Sorry, can you just say that again for me?
Sure, absolutely. How much assay development needs to be done for different lipids?
So yeah, we would recommend, as with any protocol, to run an optimization when major parameters such as lipids are changed, mostly because you are quite likely to identify something that is actually going to be changing about your formulation that you would not be able to detect by conventional methods.
Excellent, excellent. Next question I have for you, and thank you to our audience. I see all those questions coming in. We're going to try to get through as many as we can, so definitely do keep them coming. The next question that I have says, hello, thanks for the presentation. What is the lowest size limit detectable using NFC?
Yeah, so this is going to be 40 nanometers based on the scattering of light. We can also use fluorescence, and so the lower limit of fluorescence detection can be as low as a single PE unit. When we're doing labeling of mRNA copies, the smallest length of RNA that can be detected past the laser is approximately 800 nucleotides, maybe 1,000 nucleotides.
Excellent, excellent. Thank you. Next question I have, did you ever try quantifying pDNA in single particles?
Quite possibly, but what I would say is that a lot of the nucleic acids can be labeled with different methods. So if we haven't done it already, I'd be more than welcome to try it.
Excellent. Excellent. Next question I have, can we multiplex mRNA per particle?
Yes, we actually have a lot of really cool data that I'm looking forward to sharing in future webinars. But essentially, all we have to do is these kind of like this can be for either bivalent vaccines or whatever whatever is needed. There is pre-labeling that's required in this case. So we just need to take either a red or a green dye, conjugate it or incorporate it into the different RNA populations, and then you'll be able to identify the particles which have successfully co-isolated both of these RNAs.
Excellent, excellent. Next question I have, how did you measure the absolute number of ligands on the particle? And then there's a second question, is there any standard particle?
So this is a good question. So in terms of standard, like reference materials are trying to be produced by different networks. And obviously we'd like to be part of those measurements in the future. In terms of actually doing the quantification, this is based on very well-documented quantitative flow cytometry approaches. So we use an ERF or like NESF bead, which allows us to convert the fluorescent units of the instrument itself into units representing number of dye molecules. Now, with the approach we've shared today, those dye molecules are directly conjugated in single units to our target. In other approaches, those fluorophores might be conjugated to an antibody, which would then obviously bind to a target. And at that point, you'd have to know the approximate number of those fluorophores that are on the antibodies, otherwise known as the FP ratio.
Excellent, excellent. Thank you. We have lots of questions. I actually want to back up and ask a few that I had come in earlier. Does NanoFCM have a strategy to distinguish free mRNA and mRNA attached on the LNP surface?
Yes, yeah. So there's a couple of ways that we approach this. So we've talked about the use of membrane permeable dyes that interact with the nucleic acids. So because this approach labels all the RNA in the system, we could use a different dye which is non-membrane permeable, and obviously this would only attach to either free RNA or RNA that is adhered to the particle surface. Alternatively, we can digest away the RNA and measure the subtracted fluorescence once the exterior RNA and free RNA is removed.
Excellent, excellent. And another question I have here, what are the rules of selecting mRNA labeling dyes?
So we like to use things that are very robust. So we trial quite a few of these, but they're all freely commercially available. We don't create them ourselves. They're usually quite cheap, which is useful. But we look for membrane permeability. We look for even distribution, so that when the RNA copies go past the laser, they will have very similar fluorescence. Other than that, things that are bright and things that are in very, very different colors mean that the protocols we can employ can be nice and flexible.
Excellent, excellent. Next question I have, what is the size detection limit of nano-FCM for cell-free nucleic acids?
Yeah, so this would, I would say, down to about 800 nucleotides, maybe 1,000 nucleotides in length, with some of our most common fluorescent dyes. But we can use other like fluorophore conjugation methods to allow us to go even further down. But the most common method, which uses Cyto 9, means about 800 to 1,000 nucleotides.
Excellent, excellent. Next question I have, for sample processing, what is the total time and can the system be automated?
I'm trying to think the number of samples that I ask my team to go through in a day. So the processing, as we talked about, a lot of that data comes through from a single sample. When we introduce the sample into the system, and analyze it and then clean the system before the next sample. That total process takes about 3 minutes. The system is in the process of being automated, so we're rolling out the next version of the software that's been requested by users currently. What this allows for is for a carousel unit to continue loading of up to 20 samples, so allowing for basically 20 of these samples to be run in an hour's time. And obviously there's a lot of data that then can be processed from that.
Excellent. Excellent. Thank you so much. And just to remind our audience, we're taking questions. So if you have a question, at any time, you can go ahead and enter it. We'll try to get through as many questions as we possibly can in the time that we have left. Ben, the next one I have for you, whether the let me rephrase the question a little bit. Is the NanoFCM only for lipid-based NPs, or can it also be useful for other particles like polymer/proteins?
Yes, we actually get these types of particles more and more often now that people are starting to recognize the different types of nanomedicine, nanotherapeutics we can work with. So absolutely, we work with more protein-based particle formulations that sort of interact with siRNA and things like that. To date, we found very few nanoparticles that we can actually work with. So I would definitely investigate with us if yours is suitable.
Excellent, excellent. Next question I have: what are the limits of detection of the different fluorescence filters?
I think that one might require a bit too much going into. So I mean, if I start to think about some of the fluorophores that we can detect, it would be a single PE molecule. So we can obviously very low requirement there. For some of the maybe green channel and red channel, if I was to think of electrophorate 8 and 647, we're looking down to about 8 copies of those. So in terms of surface ligands that are needed, very low quantities, particularly with antibody binding, which can take up to sort of 4 or 5 of these fluorophores to a single antigen. And again, with mRNA length, we're usually looking at about 800 to 1,000 nucleotides as the lower limit.
Excellent. And this question: can plasmids be fluorescently labeled and detected into single particles? Do you already have a protocol in place? So 2 questions there.
Yes, but nice, easy questions. Yes, they can. And yes, we do. So reach out and we'll get in touch.
Wonderful. Wonderful. Ben, I really appreciate you taking time today to answer questions and also for the great presentation. Thank you so much for that. To our audience, thank you for all those wonderful questions. I'm going to bring the webinar to a close, but before I do that, a reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So please do keep an eye on your email. You should get a link within that time frame. You can come back and watch the entire presentation all the way through this Q&A. And you can also share the link with your colleagues who might not have been able to join us today. Ben, once again, thank you so much for being here and for your presentation. I'd also like to thank today's sponsor, NanoFCM, for sponsoring today's event. And lastly, I would like to thank the Gen Multimedia Group for hosting. So thank you, David Mosley, Reynolds Gutierrez, and Natalie Torn. And last but not least, thank you to our audience for being here. For everyone at Gen, I'm Uduak Thomas. Thank you and goodbye for now.